Hi everybody, and welcome back to Principles of Cell Biology. I'm your professor, Dr. Kaylin Fogelman, and today we will be covering our second chapter in this course, the molecules of cells. The molecules of cells will be broken up into two separate lectures, the first of which I will cover in this video, and this is where we will discuss the chemistry of life. We're going to be going over basic chemistry concepts that you will need as a foundation for the rest of the material in this course. It is going to cover the topics of introduction to chemistry, molecules and compounds, the chemistry of water, and organic molecules. The second part of the molecules of life chapter will come in a separate video, but this is going to cover the biological macromolecules that are carbohydrates, lipids, proteins, and nucleic acids that serve as the building blocks of life. To start on our Chemistry of Life lecture, we're going to begin with Introduction to Chemistry. So once we cover this section, we should be able to describe how protons, neutrons, and electrons relate to atomic structure, understand how to interpret the periodic table of elements, explain what is meant by the term isotope, and identify the beneficial or harmful uses of radiation. So let's begin with matter. Matter is defined as anything that takes up space and has mass, and it can exist in four states: solid, liquid, gas, and the lesser known that you hear of less commonly, plasma, is the fourth state of matter. Whether we're talking about a cell, a rock, or a glass of water, all matter is composed of elements. An element is a substance that cannot be broken down into similar substances with different properties. For example, oxygen and hydrogen are both elements, but each has unique chemical and physical properties such as their density, their boiling point, their solubility, and their chemical reactivity that define how they will interact with other elements. There are 6 key elements that are essential for life. These are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. These elements form the building blocks of biomolecules, and they make up about 95% of the body weight of organisms. The properties of these elements, specifically these 6, are essential to what makes cells and organisms unique. There are other elements that are important to organisms, things like sodium, potassium, calcium, iron, manganese, but those are found in much smaller quantities, and they're not universally needed in all organisms, such as these 6 elements here. Next, we're going to look at atomic structure. An atom is the smallest unit of an element that still retains the properties of that element. Atoms are made up of 3 subatomic particles: the proton, which is positively charged, the neutron, which has no charge, and electrons, which are negatively charged and orbit the nucleus. So the nucleus is the center of an atom. And again, this contains the protons and the neutrons within this nucleus, and the electron is going to orbit around the nucleus in specific energy levels that we call electron shells. Each element will have an atomic symbol, such as C for carbon or H for hydrogen. And each element, in addition to having an atomic symbol, is also defined by its atomic number. So the atomic number is the number of protons that each specific element has in its nucleus. For example, hydrogen has an atomic number of 1 because it has 1 proton, and carbon has an atomic number of 6. This number will never change for a specific element, and it is one of its defining properties. We also have a mass number. The mass number of an atom is the sum of its protons and neutrons. This value can vary between atoms of the same element, giving rise to what we call isotopes. This is similar to atomic mass, which you may see on periodic tables, where atomic mass does not have an even number like the mass number has, because the atomic mass is going to reference the average mass for all isotopes for an atom that are found. Well, when you see the mass number given, generally with an atomic symbol, this is the number of protons and neutrons that an element has in a specific instance or example, whereas the atomic mass is the larger value that you would find associated with an element on something like the periodic table, because this is an average of all isotopes that are existing that you would find in the wild. We will discuss isotopes shortly and dig a little bit deeper into how we determine mass number from the number of protons and neutrons that an element has in its nucleus. The periodic table, which we've mentioned several times, is a critical tool for understanding elements. Within the periodic table, we organize elements based on their atomic structure and chemical properties. Each element is represented by its atomic symbol, its atomic number, as well as its atomic mass. For instance, the symbol for hydrogen is H, its atomic number is 1, meaning that it has 1 proton in the nucleus, and its atomic mass is approximately 1, meaning that it rarely has neutrons. It just has that proton existing in the nucleus. For another example, we could look at carbon here. Atomic symbol is C for carbon. Its atomic number is 6, meaning that it has 6 protons in the nucleus always, and its atomic mass is 12.01. So this means that carbon is existing as an isotope, primarily carbon-12, with 12 protons, 12 neutrons, but sometimes there are isotopes that have more neutrons present, which is why this is not exactly an even number. Our periodic table is arranged into rows that we call periods, and then they are arranged into columns called groups. Elements within the same column or the same group have similar chemical properties because they share the same number of valence electrons or electrons in its outer energy ring, which we will discuss shortly. But all elements in group 1, for example, such as sodium and potassium, are highly reactive because they have 1 valence electron in their outer shell. And we're going to go forward discussing electrons and see more what this means. But what the big takeaway from the periodic table is that this is not organized randomly. Things are ordered and organized based on chemical properties and reactivity. We've mentioned isotopes, and isotopes are variations of an element that have the same number of protons but a different number of neutrons. While we cannot change the number of protons in an atom's nucleus without changing what that element is, we can change the number of neutrons, as a neutron has no mass and no charge. For example, carbon has isotopes such as carbon-12, carbon-13, and carbon-14 with a mass number of 12, 13, and 14 respectively. For these 3 isotopes, they will always have 6 protons. If we change the number of protons, we change what that element is. For example, if we had 5 protons, that would be boron. If we had 7 protons, that would now be nitrogen. But carbon-12 has 6 protons, and we determine the number of neutrons. The mass number is protons plus neutrons. So 6 protons, 6 neutrons adds up to 12. Here in carbon-13, we still have 6 protons, but if we take 13 minus 6, that is 7 neutrons. And then for carbon-14, that's 6 protons and 8 neutrons. Some isotopes are unstable and radioactive, meaning that they emit energy as they decay into a more stable form, but there are radioactive isotopes, or radioisotopes, which can be useful in several fields. So for example, in medicine, radioactive isotopes like iodine-131 is used to diagnose and treat thyroid disorders, where in archaeology, carbon-14 dating can allow us to estimate the age of ancient artifacts based on the way that this carbon-14 isotope decays and gives off energy as it goes back to other forms of carbon isotopes. However, exposure to high levels of radiation from radioactive isotopes can damage living cells. This can lead to conditions such as radiation sickness or an increased cancer risk. So it's important for us to distinguish between low or high levels of radiation. Low levels of radiation, like what you get from an X-ray or background environmental radiation, is generally harmless if your exposure is limited. But high levels of radiation, such as from a nuclear disaster, if we think of Chernobyl or Fukushima, or a nuclear accident, can be extremely harmful. These high levels of exposure to radiation can damage DNA, it can cause mutations, and it can lead to cell death. So radiation has dual roles. It can both benefit and harm. For example, if we harness radiation and we use it therapeutically, we can treat things like cancer as we use it to kill carcinogenic cells. But overexposure or targeting of this high level of radiation to things that aren't cancerous can cause severe health problems and kill healthy cells. Now that we've discussed an atom's protons and neutrons, we'll examine the electrons. So while the protons and neutrons exist in the nucleus, protons being positively charged, neutrons being neutrally charged or having no charge, the electrons are negatively charged particles that orbit outside of this nucleus. They're arranged in energy levels, or what we call shells. So the first shell that's closest to the nucleus can hold up to 2 electrons. If an element has more than 2 electrons, then we keep forming shells that are outside of one another. So while the first shell holds 2, the second shell and subsequent shells hold a maximum of 8 electrons each, following the octet rule. So 2 shells first, then we build shells out, filling up to 8 in those subsequent. If we fill all 8 electron slots, then we build another shell. And our octet rule states that the valence shell, or the outermost shell, is most stable when it has 8 electrons. So that valence shell determines how an atom interacts with others. Atoms that have an incomplete valence shell, meaning that they have less than 8 electrons, are reactive because they tend to gain, lose, or share electrons to achieve stability by getting that full valence shell. For example, here with hydrogen, we have 1 electron in its valence shell, and it would like to have 2 to fulfill or make this first shell full. So hydrogen will commonly pair with another atom so that we fill that valence shell. Carbon has 6 protons and electrons, so it has 2 electrons in that first shell and then 4 electrons in the second shell. And carbon will therefore want to bond with up to 4 other things to satisfy that octet rule and have a full valence shell. This is related to our periodic table organization, where all groups have the same number of valence electrons and will therefore want to interact with other atoms similarly. Valence electrons are very important for us to understand how an element will undergo chemical reactions and interact with the elements around it. Looking at some further examples of Bohr models here, the way that these electron shells are depicted around the nucleus of specific atoms, we've already covered hydrogen and carbon, and now we'll look at nitrogen. The atomic number of nitrogen is 7, meaning that it has 7 protons and 7 electrons when it has a neutral charge. It has 2 electrons in the first shell, which to fulfill the 7 electrons, we then have 5 electrons in this outermost valence shell. Therefore, nitrogen is generally going to want to gain or share 3 electrons so that it has 8 total in this valence shell. Oxygen's atomic number is 8, so it has 2 in this first shell and then 6 in its valence shell. It's going to want to gain or share 2 electrons to fulfill the octet rule. Phosphorus's atomic number is 15. It's in the same group on the periodic table as nitrogen, meaning that it has 5 valence electrons and will want to react very similarly to how nitrogen would react. And sulfur is in the same group as oxygen and also has similar valence chemistry and reactivity. Let's walk through some practice examples of identifying the numbers of protons, neutrons, and electrons of given elements. If we have the atomic symbol C, an atomic mass of 13, this is carbon 13, and we must identify the number of protons, neutrons, and electrons that it has. So first, carbon is always going to have 6 protons. If it has 5, it's no longer carbon, it's boron, and if it has 7, it's now nitrogen. So we depict that by first looking at our periodic table and identifying the atomic symbol C, and then looking at the atomic number. That is always going to stay the same. So the number of protons for this example is 6. The atomic mass, which is what is given here, is the number of protons plus neutrons. And we know the number of protons is 6, so that tells us this is an isotope with 7 neutrons. And we determine that by taking the atomic mass minus the number of protons, or our atomic number, that gives us 7 neutrons. Electrons will always be equal to the number of protons if there is a neutral charge. So if there's a charge denoted, then we have to calculate the number of electrons. But if there's no charge here, if this doesn't say that it's positive something or negative something, that means that our electrons are going to equal our number of protons. And in this instance, this has 6 electrons. Now we must determine an isotope when given the number of protons, electrons, and neutrons. So first we can determine the atomic symbol from the protons. It tells us that there are 16 protons. So we go to our periodic table and we look at which element has an atomic number of 16, and that is S, or sulfur. So our atomic symbol is S, sulfur. Then we can determine the mass number by adding the protons and neutrons. So if we add 16 protons, 16 neutrons, our mass number is going to be 32. Then we can determine the charge, which will always go on the top right-hand side of our atomic symbol. So we calculate the charge by taking the protons, which have a positive charge, and subtracting the electrons that are negatively charged. So if we have 16 protons and we have 18 electrons, that gives us 16 minus 18 is negative 2. And we can also determine this by saying we have 16 things that are positively charged and we have 18 things that are negatively charged. So we have 2 more negatively charged things. So sulfur is going to have a charge of minus 2 in this case. So we have sulfur with a mass number of 32 and a charge of negative 2. Walking through another example, we can figure out our atomic symbol by looking at our protons, which is 26. If we go to where 26 is on the periodic table, this is Fe, or iron, is what the element is here. We can figure out our mass number, or atomic mass, by adding our protons plus our neutrons. So 26 plus 32 is going to be 58 up here in the top left. And then we can figure out our charge by adding our protons and our electrons, or subtracting our protons and our electrons. So 26 minus 24, that gives us a charge of plus 2 because we have 2 more positive charged things than we do negative charged things. Here we have bromine, so we can calculate the number of protons by looking for bromine on the periodic table, and it is here, Br. So we have 35 protons. And then for neutrons, we take our atomic mass, 79 minus 35, and that is going to give us 44 for our number of neutrons. And then for our electrons, it has a charge of plus 1, which means we have to have 1 more proton than neutron, or 1 more proton than electron. So that gives us 34 electrons. And here are some practice problems for you to work through. If you'd like to pause the video and work through them, or come back to these, you can always put the answer in the chat if you'd like. But this will help you practice some of these problems of determining what an element is, the number of protons, neutrons, and electrons, or determining the mass number. This brings us to the end of our first section in the chemistry of life portion of the molecules of cells. So by now we should be able to describe how protons, neutrons, and electrons relate to atomic structure. We should also be able to identify the number of protons, neutrons, and electrons of a given element should we be given an example. We should be able to understand how to interpret the periodic table of elements, explain what is meant by the term isotope, and also identify the beneficial and harmful uses of radiation. This now brings us to our second section, molecules and compounds, where after visiting this, we should be able to describe how elements are combined into molecules and compounds, list the different types of bonds that occur between elements, and explain how hydrogen bonds are formed between molecules. So let's begin with the distinction between molecules and compounds. A molecule is formed when two or more atoms of the same element bonds together. For example, oxygen gas, depicted here, is a molecule made up of two oxygen atoms. So here we have O2. A compound, on the other hand, consists of two or more different elements bonded together in fixed proportions. Water, or H2O, is a compound made of hydrogen and oxygen. Elements, except for noble gases, typically form bonds because their atoms are more stable when their outer electron shells are full. These bonds allow elements to combine and create molecules that are essential for life. Noble gases aren't reactive because they already have a full valence shell and satisfy the octet rule. So if we look at noble gases that appear in this last group in the periodic table, helium, neon, argon, krypton, xenon, they all already have full valence shells, which tells us that they do not want to react or give up or gain any electrons. So in this section, we're going to look at different types of chemical bonds and how they form, and a lot of this has to do with their valence chemistry. Chemical reactions occur when two or more atoms bond together to form a compound or a molecule, or if molecules are breaking apart into smaller components. During these reactions, energy is either going to be released or absorbed due to the breaking and forming of chemical bonds. The chemical bonds that are formed between atoms include ionic, covalent, and hydrogen bonds. So these bonds determine the structure and behavior of the molecules that they form. Ionic bonds are formed when electrons are transferred from one atom to another, creating charged particles called ions. Covalent bonds are formed when two atoms share electrons to achieve stability or fulfill that outer valence shell's octet rule. And hydrogen bonds are a specific type of bond where weak attractions between slightly positive hydrogen atoms and slightly negative atoms in a nearby molecule or a region of the same molecule will be attracted to each other based on those partial positive and negative charges. So each of these bonds are going to play critical roles in the structure and function of biological macromolecules, so we're going to look at them in more detail. An ionic bond is formed through the transfer of electrons between atoms, resulting in charged particles known as ions. So an ion is formed when an electron is transferred from one atom to another, and then those atoms then have either positive or negative charges. So when we create ions, we can have a positively charged ion, a cation, or a negatively charged ion, an anion. So a cation gets a positive charge from losing an electron. In this example here, sodium has one electron in its valence shell, which makes it quite unstable, and it wants to give up that electron. When it gives up that electron, it then has an overall positive charge because it has one more protons and electrons now. Chloride, on the other hand, has 7 electrons in its valence shell, and it wants to gain an electron, such as that one that sodium just donated. So when chlorine accepts a donated electron, it now has a net 1 charge, negative electron charge, because it has one more electron than proton. So cations have positive charges, anions are ions that have negative charges. And an ionic compound is a compound that is formed because two things are held together by an attraction of those ions. This is called an ionic bond. So looking at a specific example of this with sodium chloride, this is what you know as table salt. So sodium has 1 electron in its valence shell, chlorine has 7 electrons in its valence shell. They're both unstable because of this. So sodium wants to get rid of this electron, chlorine wants to gain an electron. So sodium donates this electron, which will make it have a +1 charge, and chlorine will accept an electron having a -1 charge. Now this is positively charged and this is negatively charged, and they are attracted to each other, kind of like how a magnet is attracted to another magnet. So the positive and negative charges of these ions is what forms that ionic bond and creates the ionic compound. We also have covalent bonds, which form when two atoms share electrons to fill their outermost shell. Instead of donating like ions, they're going to share the electrons and come together, kind of like they're holding hands. So instead of giving up or receiving, they're going to join together and be held together because they're sharing the electrons in their valence shells. So these shared electrons allow each atom to achieve a stable configuration and follow the octet rule. For example, in a molecule of water, like H2O, each hydrogen atom is going to share an electron with the oxygen atom, creating a covalent bond. So covalent bonds are often represented using structural formulas like HOH for H2O or water. And a lot of the way that these molecules and compounds are depicted is in a linear shape or a flat shape. So if we have H2 or O2 or CH4 here, methane, many of the ways that we depict these show them as looking like they are flat. But these actually exist in 3D space. They're three-dimensional unless they're linear. So in the instance of H2 or O2, if they have two atoms, they are linear. But if they have three or more atoms, these exist in three-dimensional shape, such as methane here, CH4. You have carbon in the middle, and then it makes this pyramid shape. It's a tetrahedral with 4 hydrogens, making that pyramid around the carbon. So it's important for us to understand that even though we will see these molecules mostly represented as two-dimensional structures, that they do exist in 3D space, and our ability to visualize that 3D space and those 3D structures will help us to understand the biological function and interactions of these molecules. So looking more at our types of covalent bonds, we can classify these as either non-polar or polar covalent bonds, depending on how equally the electrons are shared in the molecule. So a non-polar covalent bond occurs when the sharing of electrons between two atoms is equal. For example, in oxygen gas, or O2, both oxygen atoms pull on the shared electrons equally. The same here if we have H2. These electrons are spending equal amount of time orbiting around the different nuclei, the same as we would see here with CH4. None of these hydrogens are getting the electrons any more than any of the other hydrogens, and the electrons are equally distributed around this central carbon. Then we have polar covalent bonds, which occur when one atom is exerting a stronger pull on the shared electrons. So polar meaning that it has, you can think of that as polar has a pull to it. So when there is a stronger pull of one of the atoms in the molecule, that part of the molecule has a higher electronegativity, and this causes an uneven distribution of the charge. So a great example of this, of a polar molecule, is water. Here you can see that this is not as evenly distributed as the H2 or CH4 examples that we gave for non-polar covalent. Here, oxygen has more electronegativity and it's pulling the electrons closer to itself. There are more electrons spending more of the time orbiting the nucleus of this oxygen than the hydrogens. So this results in a partial negative charge, depicted here, on this oxygen. There are less electrons orbiting around these hydrogens, which causes them to have less electronegativity or a partial positive charge. So the way that I think about whether something is nonpolar or polar covalent, if this helps you, is when I look at a nonpolar covalent molecule, to me it looks balanced. It looks equal, it looks evenly distributed. But when I look at a polar covalent molecule, it looks unequal or unbalanced to me. I see that it's leaning more in one direction or the other as to where the electrons are spinning the majority of the time. And this brings us to hydrogen bonding, which are weak but crucial for many biological processes. Hydrogen bonds occur when a hydrogen atom with a partial positive charge, due to the molecule's electronegativity, is attracted to a nearby atom with a partial negative charge, often oxygen or nitrogen. So for example, in water molecules, hydrogen bonds form between the slightly positive hydrogen of one molecule and the slightly negative oxygen of another. So if we see water molecule here, H2O, with the oxygens being red and the hydrogens being this tan or gray color, our hydrogens all have a slightly positive charge and our oxygens all have a slightly negative charge. So the hydrogens in one water molecule are going to be attracted to the oxygens in another water molecule because all of the hydrogens have a partial positive charge and all of the oxygens have a partial negative charge. So every hydrogen is going to be attracted to oxygens in other parts of the molecule. Hydrogen bonding is weak individually, but the collective strength of hydrogen bonds with many molecules put together is quite significant. Hydrogen bonding is what gives water its unique properties, such as high cohesion and the ability to dissolve many substances. And this hydrogen bonding also plays a critical role in stabilizing the structure of macromolecules, such as proteins and DNA. So in this section, we've explored how atoms combine to form molecules and compounds, the types of chemical bonds that hold these structures together, and the unique properties of ionic, covalent, and hydrogen bonds. These concepts are foundational for understanding the chemical interactions that drive biological processes. In the next section, we're going to look at the properties of water and why it's essential to life. We will also look at what values on the pH scale indicate about a solution and describe how buffers are important for living organisms. So starting out with the properties of water, water is the most abundant molecule in living organisms and is absolutely essential for life. Its unique properties stem from its molecular structure and the hydrogen bonding between water molecules. So let's take a look at why this is so significant. Water exists in three states: solid, liquid, and gas under the natural conditions on Earth. So in its liquid state, water molecules are constantly breaking and reforming hydrogen bonds. This is what gives water its fluid nature. This is the state most relevant for biological processes. But we also have a solid state where water forms a crystalline lattice, with each molecule hydrogen bonded to 4 other water molecules. This structure spaces the molecules farther apart, and this will contribute to making ice less dense than liquid water, a property that we're going to talk about is critical for aquatic ecosystems. In the gaseous state, water molecules are no longer bound by hydrogen bonds and exist as individual molecules. So this is what forms water vapor in the atmosphere. Water's freezing and boiling points are also key to life. So it freezes at 0 degrees Celsius and boils at 100 degrees Celsius under standard atmospheric pressures. But these temperatures are relatively high compared to other similar sized molecules thanks to this hydrogen bonding. Without hydrogen bonding, most water would exist as a gas at most temperatures on Earth, making life as we know it impossible. The solid to liquid and liquid to gas transitions require significant energy. So these transitions, and because they require a lot of energy, moderate temperature changes in the environment. This protects organisms. Hydrogen bonding is key to the way that water behaves. So the water molecules, as we've seen, they have a bent shape with 2 hydrogen atoms covalently bonded to a central oxygen atom. Because oxygen is highly electronegative, it's going to pull electron closer, creating slight negative charge on the oxygen and slight positive charge on those hydrogens, as we've seen. This polarity of the water molecules is what enables it to form hydrogen bonds, those weak interactions between positively charged hydrogens of one molecule and the negatively charged oxygens of another. These bonds are weak. We can easily break one of them, but when you put many of these bonds together, such as in a volume of liquid water, they create significant stability. This is responsible for many of these properties of water that we're going to discuss that support life. So there are 5 key properties that make water vital to life. Many of these properties are due to this hydrogen bonding we've been discussing. So first, water has a very high heat capacity. Second, it has a high heat of vaporization. Third, it acts as a solvent for many substances. Fourth, water molecules are cohesive and adhesive. And fifth, frozen water is less dense than liquid water. Each of these properties play an important role in maintaining the stability of living systems and their environments. We're going to discuss each of these in detail. So first, starting out with water's heat capacity. Water's high heat capacity is one of its most important properties for life. But what does this mean? So heat capacity refers to the amount of energy required to raise the temperature of a substance. For water, this capacity is unusually high due to the hydrogen bonds. So each time water absorbs heat, much of that energy is used to disrupt these bonds rather than immediately increasing the movement of water molecules. A critical measure of heat capacity is the calorie. So a calorie is defined as the amount of heat energy needed to raise the temperature of 1 gram of water by 1 degree Celsius. So when you hear calorie of your food, that is actually an energy measurement. So it's talking about the amount of heat energy required to raise the temperature of 1 gram of water by 1 degree Celsius. So calories in your food are not inherently bad. Calories translate to energy. So calorie being 1 gram of water increased by 1 degree Celsius, and this sounds small, but when applied on a big scale, it explains why water is so effective at regulating temperatures in both organisms and the environment. So compared to other substances, water requires almost twice as much energy to raise its temperature. To give an example, metals like iron heat up really quickly because they have a much lower heat capacity and they don't have hydrogen bonding. The high heat capacity of water allows water to act as a thermal buffer, meaning it resists rapid temperature changes. And this is really biologically important because for organisms, water's ability to stabilize temperature protects them from sudden changes in their internal and external environments. If we have large bodies of water, such as an ocean or lake, these large bodies of water can absorb and store heat from the sun during the day and release it slowly at night. This helps keep coastal climates relatively mild. And inside our bodies, the water in our blood and our tissues and our cells absorbs heat from metabolic reactions, and this prevents dangerous spikes in our body temperature. Aquatic ecosystems really benefit from water's high heat capacity because it ensures that the temperatures of lakes or rivers or oceans change slowly, allowing organisms to adapt to temperature changes over time where we're not seeing rapid fluctuations in temperature like we might see in the air from day to day or season to season. So our second property is water's high heat of vaporization. And this is another property that is crucial to water's role in life. So heat of vaporization refers to the amount of energy required to convert 1 gram of liquid into gas. For water, this is exceptionally high because all the hydrogen bonds in water molecules between each individual water molecule has to be broken for the molecules to escape as vapor. On average, water requires 540 calories to vaporize 1 gram at its boiling point of 100 degrees Celsius. This is important for several reasons, several biological implications here. Animals are able to cool through evaporation. When water evaporates, it takes a large amount of heat with it. This is why sweating is such an effective cooling mechanism for humans and animals. As sweat evaporates from the skin, it's absorbing heat, reducing body temperature. And for animals that can't sweat or don't sweat well, they have other adaptations. So for example, swine will roll in mud or puddles so that they get a coat of liquid on their body, and it acts similarly to sweating activity. And other animals like chickens or dogs will pant. for a similar mechanism of trying to evaporate liquid to pull heat away with it. Another important biological implication is climate moderation. So large bodies of water, like oceans, absorb heat during the day and release it as vapor. This then condenses into rain. So this process helps stabilize global temperatures. In plants, transpiration uses water's heat of vaporization to cool the plant and maintain a steady flow of water through its tissues. And then on to our third property. Water is often called the universal solvent because of its ability to dissolve more substances than any other liquid. This is due to water's polarity. So the slightly positive hydrogen atoms and the slightly negative oxygen atoms attract and surround ions or polar molecules, breaking them apart and dispersing them in a solution. So let's consider the components. Solutes are substances that dissolve in water, such as salts or sugar. Here in this example, we have NaCl, sodium chloride or salt. A solution is the homogeneous mixture of the solutes in the water, and the water in this case being our solvent, or the thing doing the dissolving. Substances that easily dissolve in water, like salts or sugars, are called hydrophilic or water-loving, and they interact because of water's polarity and the fact that they also have polarity. Non-polar substance, they do not have polarity, they don't have any partial charges or full charges anywhere on them, are hydrophobic. They are afraid of water. That's what that hydro and then phobic. They do not dissolve in water, and it's because they are not polar and they do not want to interact. So an example of this that you'll be familiar with is a fat or an oil. So if you put oil in water, the water is polar, the oil is non-polar, they do not want to interact with each other. The solvent property of water is essential for life because it allows organisms and the cellular processes within those organisms to do a variety of things, such as it allows cells to maintain homeostasis by dissolving nutrients, ions, or gases. They can be transported throughout the organism, and it enables a lot of biological reactions that sustain life, as reactants must dissolve in water to interact effectively. Water's ability to stick to itself is known as cohesion, and its ability to stick to other substances is known as adhesion. Cohesion and adhesion are another property that support life. Cohesion is due to hydrogen bonding, where water molecules are attracted to each other. This property gives water a high surface tension, which allows small organisms like insects to walk on water, as depicted here. And it also plays a crucial role in creating the spherical shape of water droplets. And this is because those water molecules are all attracted to one another. Adhesion occurs because water molecules can stick to polar or charged surfaces. So for example, water adheres to the walls of plant vessels, helping it travel from the roots to the leaves and be transported to where it needs to go within a plant. The two properties together of cohesion and adhesion are what enables capillary action. So this is where water moves through a narrow space against gravity, and capillary action is critical for processes such as water transport in plants via the xylem, or blood flow in tiny capillaries within the human body. And on to our final property. Unlike most substances, water becomes less dense when it freezes, which is why ice floats on liquid water. So when water cools, its molecules slow down and arrange themselves into a crystalline structure that's held together by hydrogen bonds. This structure spaces the molecules farther apart than in liquid water, which contributes to the density. This property is critical for life on Earth because it creates insulation. Ice forms a layer on the surface of lakes and ponds, insulating the water below and preventing it from freezing solid. So bodies of water are freezing from the top down, and when we start to have a layer frozen on the top, it insulates everything below from the external environment. This allows aquatic life to survive in cold climates. It creates stable habitats for organisms that rely on ice sheets or shelves for survival for habitat, and for things underneath that wouldn't be able to survive if that body of water was frozen solid. Without this unique property, many aquatic ecosystems would not exist as we know them. And you may be wondering why we keep bringing up the properties that affect large bodies of water and the aquatic organisms within them. And that's because the majority of planet Earth is water, and the majority of living organisms live within those bodies of water. So when we talk about properties that are unique to or sustain life on Earth, we need to consider all life on Earth in both terrestrial and aquatic environments. Now that we've discussed the properties of water that are important for life, we're going to discuss another important aspect of the chemistry of water, which is acids and bases. So water can ionize, breaking into hydrogen ions, or H+, and hydroxide ions, OH-. So H2O, when we break that up, we get one H+, and then an O, and then the other H with the O, and that's OH- or hydroxide ion. So the property of water ionizing into hydrogen ions and hydroxide ions acts as a medium for acids and bases. So acids are substances that release hydrogen ions when they dissociate in water, and this means that a solution has a greater hydrogen ion concentration than hydroxide ion concentration, more H+, essentially. Acids have a sour taste and they can corrode metals. Examples of acids include vinegar, lemon juice, coffee, tomatoes. HCl is an example of an acid, hydrochloric acid, and when this ionizes or dissociates in water, we're left with hydrogen ion and chloride ion. So because we have more H+ than OH-, this is an acid when it ionizes. The opposite is basic solutions. So basic solutions have a low hydrogen ion concentration, or essentially they have more hydroxide ions than hydrogen ions. So bases are slippery to the touch generally, and they have a bitter taste. Examples of bases include baking soda and ammonia. Acids and bases are both essential for biological processes. Like stomach acid, the HCl that's shown here, helps to break down food, while bases in the small intestine neutralize that acid to prevent damage as things pass through the digestive tract. An example of a base, sodium hydroxide, when it ionizes, we're left with sodium and OH-. In this example, you can see how we have more hydroxide ion than hydrogen ions. The pH scale measures how acidic or basic a solution is, and this is based on the concentration of hydrogen ions. Anytime we see these square brackets around something, that's read as concentration of. So square brackets around H+ is saying this is the negative log of the concentration of hydrogen ions. So our pH scale, which is telling us about the acidity or alkalinity of something, ranges from 0 to 14. If the pH is lower than 7, that substance is acidic, and that's telling us there are more hydrogen ions than hydroxide ions. If the pH is 7, that is neutral. We have an equal amount of hydroxide ions as hydrogen ions. And then if the pH is greater than 7, going up to 14, that is a basic or alkaline solution, meaning that we have more hydroxide ions than hydrogen ions. Each step on the pH scale represents a 10-fold change in hydrogen ion concentration because it is a logarithmic scale. So for example, a solution with a pH of 5 is 10 times more acidic than one with a pH of 6. Biological systems rely on maintaining very specific pH ranges. As we'll see in this next slide, our blood needs to exist within a very narrow range of pHs. Blood pH is approximately 7.4. If it goes below that, that's considered acidosis. If it goes above that, that's considered alkalosis. We have buffers in our body, buffer systems, that help pHs resist change. So our body does not want to change from those pHs that are doing very specific roles in different parts of the bodies. And the way that these buffers work is that they are taking up either hydrogen ions or hydroxide ions to neutralize a basic or acidic substance. So a great example of the way that buffers maintain homeostasis within our bodies is the bicarbonate buffer system, which exists in human blood. So we don't want our blood to become too low of a pH or too high of a pH. If blood starts to become too acidic, bicarbonate is going to bind with excess hydrogen ions to form carbonic acid. Here we see carbonic acid, bicarbonate ion. If we start to get too many H pluses, it's going to combine with bicarbonate and that will form carbonic acid. If we start to get too much OH, our hydroxide, if our blood starts to become too basic, those hydroxides will combine with carbonic acid to form bicarbonate and water. So we have what happens when we start to become too acidic and get too much hydrogen, and what happens when we start to get too basic and get too much hydroxide. So you may look at this and say, in the instance of our bicarbonate buffer system working, taking up hydrogen ions and forming carbonic acid, isn't the point to get rid of acid? So this is what happens when our blood starts to get too acidic. If we form too much carbonic acid, well, sometimes the carbonic acid gets used to neutralize substances that are too alkaline or basic, but if we have an excess of that, this carbonic acid can dissociate into H2O and CO2, or carbon dioxide, which will just be exhaled, the way that carbon dioxide is from our respiratory system. So your body works very hard to maintain a very specific pH range in each part of your body. And in this section, we explored water's unique properties, its role in maintaining life, and how those interactions with acids, bases, and buffers help to stabilize our biological systems. These concepts underline why water truly is the foundation of life. And in our final section for this lecture, we're going to examine the organic molecules that form the building blocks of living organisms. We're going to compare inorganic and organic molecules. We'll identify the role of functional groups, and we'll recognize how monomers are joined to form polymers. So organic molecules are the foundation of life, and they're distinguished from inorganic molecules by their composition and their structure. Organic molecules always contain both carbon and hydrogen. They often include other elements such as oxygen, nitrogen, phosphorus, and sulfur, those other elements foundational to biological life, but at a minimum they have to have carbon and hydrogen. What makes carbon so special? Why does it have to be in every organic molecule? This has to do with its valence chemistry. So carbon has 4 valence electrons, meaning that it can form up to 4 covalent bonds with other atoms. This allows carbon to act as a versatile backbone for large complex molecules. So with carbon being able to bond to other carbon molecules, it can form large hydrocarbon chains. This serves as a skeleton for organic molecules. It can also form hydrocarbon ring structures. So you see here, each of these carbons can form 4 double bonds, or you can have it connected to 3 things with double bonds, so 4 bonds total here. But the ability of carbon to serve as the foundation for all organic or living molecules is this valence chemistry of carbon. And the reason it's always coupled with a hydrogen is also due to the reactivity in the valence chemistry of hydrogens as well. So these hydrocarbon skeletons and rings are going to serve as the backbone of organic molecules that are functional to life, like carbohydrates, lipids, proteins, and nucleic acids that we're going to discuss more in our next lecture. So what differentiates one carbon skeleton from another? The way that we're able to give these molecules unique properties is by adding functional groups to the hydrocarbon skeletons. So functional group is added and it gives organic molecules its unique characteristics based on what those functional groups are and where they are located. So these will influence the chemical behavior of the molecule as well as its reactivity. Where we put the functional group, its specific location on a carbon skeleton determines the properties of what that molecule is. So some common examples of functional groups include hydroxyl groups, or OH. This is found in alcohols, and it makes water molecules polar, or it makes molecules polar and water soluble. So the R here, this represents what this functional group OH is attached to. So this is a larger part of a molecule, whether it's a hydrocarbon skeleton being a chain or a ring. This R essentially depicts something like this, and it's showing that an OH is being attached. We also have carboxyl groups. These are acidic groups, COOH. They're found in acids like fatty acids and amino acids, and they act as a weak acid. We can also have amino groups. So this is where we would have an NH2 attached to a hydrocarbon or larger molecule structure. It acts as a weak base, and it plays a key role in protein structure. As you'll see, amino groups, we're going to see these nitrogen groups in amino acids, which come together to form proteins. Another one that we see a lot is CH3, or a methyl group. This is nonpolar, and it's going to influence gene expression when attached to DNA. The placement of functional groups on a carbon skeleton determines the molecule's specific properties. So if you see here, we have a carbon chain with hydrogens attached, and then we have these functional groups attached in different areas. So the only thing differentiating this chain from that chain is the number of carbons in the chain, and then the groups that are attached to it in place of just being filled with hydrogens and all of the other bonded slots. So this carbon chain has a carbonyl group, a carboxyl group, it has an amino group, and it has a methyl group. And if we moved these around, or we substituted other functional groups in their place, that would change the structure of the molecule and what this molecule ultimately is. For instance, the difference between testosterone and estrogen lies in the functional groups. What functional groups are present, and this affects their role as hormones, and essentially the whole job of those molecules just based on the functional groups, but the core structure is the same. And this brings us to polymers. So many organic molecules are large structures called polymers, which are made by linking smaller repeating units called monomers. So mono being one, poly being many. We get a polymer by putting monomers, singular units, together to form a larger chain of something. The process of making polymers, or polymerization, allows for the creation of complex and diverse macromolecules, like carbohydrates, proteins, nucleic acids, and lipids, which we're going to talk about in our next chapter. You can think of monomers and polymers as monomers being a bead or an individual unit, and when you string them together, like forming a necklace or a bracelet, they form something completely different that has a purpose. And the way that we make polymers is through a reaction known as a dehydration synthesis. So this is also called a condensation reaction, because we're taking monomers, or individual units that we want to combine together, and we're going to pull one H and one OH minus out. And if you get an OH plus an H, that gives you an H2O. So it's called a dehydration reaction because out of our monomers, we're taking out a water, or a condensation because we're pulling a water out, creating a water. So when we take out an OH minus and an H from a monomer, two monomers, that will link them together. So we'll pull out the OH minus and the H that's keeping them separate, and when we pull those out, form a water, it's going to cause them to link together. We can have the reverse happen, and that is how we break apart a polymer if we wanted the individual monomers that make it up. So the opposite reaction is called a hydrolysis reaction, and this would use up an H2O. So if we have a polymer and we want to break it apart into its individual monomers, we would put water in, break up that water into an OH and an H. Those would separate the monomers and keep them from coming back together. And that's called hydrolysis because we are adding water into that reaction. So to summarize that section, organic molecules are the building blocks of life. Carbon's versatility allows it to form complex structures, while functional groups give molecules their unique chemical properties. Through processes like dehydration synthesis and hydrolysis, monomers are assembled into polymers or broken down into those individual subunits as needed to sustain life. So that brings us through our portion covering the chemistry of life, and that sets the stage for exploring our next section, which is biological macromolecules, which we're going to cover in the next lecture. So we now have the foundation to move forward with these next topics, where we look at the specific function and structure of carbohydrates, lipids, proteins, and nucleic acids. So join us for the next lecture, where we'll cover these biological macromolecules, and thank you for following along. I'll see you in our next class.